pattern in plant development. So in this chapter, we have several learning objectives. Okay, so if you look on here, we have about 5 LO. Alright, so for the first one is make the distinction between plant growth, differentiation and development. So for the first LO, this is actually when we go through all the slide, okay, so you need to know what is the distinguish, okay, what is the difference, okay, between plant growth, differentiation and also what is the development. Second one is you need to know the nature of the plant meristems and their contribution to the growth and development. Okay, as I mentioned to you during your first lecture, if I'm not mistaken, okay, during chapter 1, okay, I told you about the anatomy of the plant and they have the part that we call as meristem. So in this LO, you will know more detail about the meristem part, okay, and what the function, okay, or their contribution to the growth and development of plant. Third LO is understand the events associated with seed development and germination. Okay, so the event is means the activity or the process, okay, of the seed and also seed development and also seed germination. Okay, fourth learning objective is know the pattern of development for the plant embryo to adult plant. So this one you may know that actually all the living organisms, they should be coming from the embryo and to the adult plant. And the last one, you will need to understand the senescence and program cell death as the final stage of the plant development. Alright, so let we look on the introduction. So for the introduction, you need to know what is the difference between the animal and plant growth and development. Okay, because animal also is the living things. Okay, so we are really actually in a same group, okay, under the eukaryote. So what difference between the plant and animal? So actually the major difference between plant and animal is about the growth and the development of the plant is not limited to an embryonic or juvenile period. Okay, but these all growth and development, okay, they occur throughout their life, okay, of the plant. Okay, if you can do some observation, the plant start from their seed, okay, and they germinate, they develop until from the young until to adult, they always produce their new leaf, they always produce their new roots, or they always produce their new branches of stem. It's different to the animal, okay, because the main part of their organs, okay, or of their physiological process is mainly or limited to their embryonic period. So that are difference. So for the typical plant, they consist of embryonic organs, developing organs, and also the mature organs. Okay, then you also will be learn about three different terms, which is what is the growth, development, and what is the differentiation. Alright, so for the development, they actually refer to all the changes, okay, that a cell, a tissue, an organ, or the organism that goes throughout their life cycle. Okay, for example, if you cannot see through the plant, okay, you can imagine yourself, which is you actually from the fetus, you have the development to the baby, and from the baby you become the kids, okay, kids to the teenager as you all, okay, and grow as an adult. So this the appearance difference, okay, we call as the development until you die. Okay, that is different to the growth. The growth is means is more targeting on the cell, means internal process. okay, which is growth is increasing number of cell or increasing size of cell. So if you look on here, they are irreversible, okay, quantitative changes, okay. It's about the cell number, okay, the size or the volume, which is not about the changes of the appearance throughout the life cycle, okay, for the plant or for the animal. Okay, and how you able to measure the growth of certain plant. Okay, you can measure it quantitatively. Okay, either you can measure their fresh weight or their dry weight. You have the last term, which is differentiation. Okay, but before we go about the differentiation, you need to know that for cell division and cell enlargement, they actually do in a separate event, means a separate process or separate mechanism. Okay, so it can be occur both or it can be occur single. Okay, singles mean it can be occur in the absence of the other. So what means by differentiation? Okay, differentiation is referred to the changes in cells, okay, tissue or organ, okay, rather than about the size. So if we look on the growth, they are increasing number of cell, okay, only cell, okay, all about the cell. Okay, the development is regarding to changes that happen throughout your life, okay, which is from the seed, for example, for the plant, okay, to their young plant until to their adult plant. Okay, differentiation means all the changes. Okay, differentiate. If in the simple words, okay, you have your cell. Okay, your cell, a group of cell, it will be differentiate as a tissues. Okay, in different tissues, okay, a tissue, it can be differentiate to the different organ. So that means by differentiation. So changes from the cell to the tissue to the organ. So because of that, the plant can have organ like root, organ like leaf, organ like their stem. Okay, this differentiation involve qualitative changes, okay, in cell, tissue and organ. Why qualitative? Because they differentiate by become a different organ that you can see. Okay, which is you can see they have the leaf, different leaf, a lot of leaf. Okay, they have a root and they have height of stem. Okay, different height of stem for the different plant. For example, for the young compared to the old. Okay, so this differentiation is different to the cell division and enlargement, which is they do not occur concurrently. Means the growth and differentiation did not occur in the same time. Okay, because for the differentiation, they require first the growth. Then after they have the growth, they may have the differentiation. They not occur concurrently. Alright, so from the differentiation, okay, they able to reverse, okay, able to reversible, okay, where the plant cell can revert, okay, to the an embryonic form. Means from the adult plant, they able to revert or they able to reverse back as the callus. Okay, so this process can only able to achieve by using the biotechnology technique using the plant tissue culture. So because here if you look on the slide, they given you a picture of two lab assistant, okay, do the something experimental regarding to plant tissue culture. So this is they isolate the cell from the plants, for example from the leaf, so it can be induced to differentiate. Just now you learned about differentiate. Now, in order to differentiate of plant, they able to reverse, okay, to an embryonic form. So they able to differentiate, okay, to producing a mass of cell that you can call callus. So from this callus, okay, you need to know the plant they have the ability to revert, okay, to the embryonic state without passing through a reproductive stage. So this ability we call as totipotency. So all of plant they have this totipotency, which is if you look on this slide, okay, they have one carrot. So one carrot they only need to get a very thin slice of carrot and they grow, okay, onto the artificial media that have the micronutrient, micronutrient, okay, and that's very thin slice become one tree carrot plant and they can produce the carrot. You can imagine if they from one carrot they able to produce 100 thin slice of carrot. So you can imagine it able to produce 100 carrot plant from only one carrot. So this all of this technique actually need to use plant tissue culture. So most plant cells are totipotent. So with the exception of highly specialized cell modified to highly to differentiate means highly specialized cell means adult cell, okay, a matured cell is able to modify, okay, to highly to differentiate means become to their embryonic mass. Okay, all of these, okay, we call as genetic program. So this genetic program actually should be always influenced by the neighboring plant cell. Alright, so we go to the plant meristem. So for the plant meristem, okay, they are the parts of plant where they are really have high concentration or high number or high rate of the cell division. So for the meristem, they have two apical meristem, okay, which is you can find it at tips of the root or tips of the stem. So the growth that are provided by this apical meristem we call as primary growth. I know all of you have been learned before for the plant they have a growth that we call as primary growth and we can also a second one we call as secondary growth. So now we talk about the primary growth. Alright, so in the meristem, they have two really popular apical meristem that you need always remember because mostly in the final exam or in test, they always asking you about the RAM and also about the SAM. So now we look what is RAM. So RAM is the root apical meristem because as I told you before for the meristem, they have at the root, they have at the shoot. So at the root, we call as root apical meristem. So this root apical meristem or RAM is a cluster of division cells that are located behind the root cap. So you need to know that all the root they have their root cap. So this RAM they have actually at the behind of the root cap. Okay, so what is the function or the role of the root cap? Actually, this root cap that they able to secrete or they able to release one gel that we call as mossy gel. In mossy gel, they consist of pectin, right? So what function of this secretion? They actually really have in order to lubricate the root tips, okay, in order they able to penetrate smoothly it through the soil and they able to improve or they able to enhance the recovery of the mineral nutrient. As we know that the root, not only the function is only to anchor or the hold the soil particle, but they are the main also in order to reach the mineral nutrient for the plant growth. Okay, and a second role of the root cap is of course for the perception of gravity. In the root cap also, okay, or in the ram, you need to know that, okay, in each of the, okay, when the ram divide, so one will remain to continue the cell division. Okay, as we know that for the meristem part, they always do divide. So one will remain to continue the cell division. And another cell that are produced from the division of the ram, it will be elongate to push the root tips through the soil. Okay, for one, of course, to get the mineral nutrient. So if you look on the diagram, okay, they have the illustration, okay, on their ram, which is, you can see they have the name or the part that we call as quiescent center. So this quiescent center actually is the area where they have a slowly dividing cell. Okay, so this one is in the center of ram. So in this quiescent center, the cell division, they produce a new root tissue. So from the cell that they are divided, they produce root tissue and also the cell for the root cap. And you can see this quiescent center, actually they are above the columella and they are below the endodermis. Okay, second meristem is SAM. So for SAM, okay, if you need to explain about their structure, you always need to mention that this SAM is more complex, okay, than the root. Why? Because they have the diversity of the cell and tissue. Okay. So this diversity of cell and tissue is only you need to explain about their structure, which is when you look on the illustration, you can see that SAM, their shape is a dome shaped structure. So their structure is like a dome, and they are organized, okay, from two parts, which is first part we call as tunica, second part, okay, we call as corpus. Okay, so for the tunica, they are the cell that undergo the anticlinal division. Okay, so this anticlinal, if you look on the illustration, I given to you two comparison, okay, which is for the periclinal division and second one is anticlinal division. So you can see that for the anticlinal division, one cell when they are divide side by side. Okay, that is anticlinal. Okay, and for the inner corpus, okay, corpus, you can see the corpus, okay, is below the tunica. So this inner corpus layer, they generate most of the shoot. Okay, and if you can see, they also have leaf primordia. Alright, so what is leaf primordia? So if you look on the tips of the shoot, okay, you can see that they have a really young leaf. So that young leaf we call as primordia. So the pattern of the primordia actually is dependent on plant species. Okay, and mostly the shape of the primordia, they are flatted shape. Okay, now we go to the two different growth of the plant. Okay, first growth we call as primary tissue. So for the primary tissue, you can see that when you cut the stem, they only have primary phloem and they have primary xylem. And this phloem and xylem, they are separated by the vascular cambium. Okay. However, okay, from young, okay, become adult or become matured plant. And you can see what difference between the young and also the matured plant is for the matured plant, they have a thick bark, okay, a thick bark. Why they have a thick bark, okay, or a thick stem? Okay, because that plant undergo the secondary growth. Okay, just now for the young they have the primary, okay, and then they have the secondary growth. So for the secondary growth, they also have vascular cambium. They also have the phloem and xylem. However, because they increasing the diameter of the bark, that is because they have the additional of the phloem and also the xylem. So instead of they have only primary, for the secondary growth, they have the secondary phloem and secondary xylem. And of course, similar, they have the vascular cambium that will be separate, okay, between the xylem and phloem. So you can see the difference in the illustration, okay, for the primary growth in stem and also for the secondary growth in stem. All right. So from the uh, meristem, okay, you need to learn about the seed development. Okay, so for seed development, of course, you need to recall back regarding to the process of fertilization. So in the flowering plant, of course, the process of fertilization should be occur, okay, by formation of the zygote, okay, from the gamete cell. So this process occur in the maternal organ of flowers. Because of that, in this part, you need to learn back about the structure of the flower. So from the subsequent growth and the differentiation of the zygote, okay, means the consequent process from the fertilization of zygote will be resulting the embryo. So this embryo, okay, then will be have the case. Okay, so the encasement of the embryo in the seed. So from the embryo, they become the seed, right? So from the seed, they will become the matured seed. So this matured seed actually will be changed the seed into the dormant state, which is when the seed they have they have been given a favorable condition. Means favorable condition means give enough water, give enough nutrient to that matured seed. So from the dormant state, it able to germinate. So they able to germinate, become one young tree. Alright, so in the last part in this video, so you need to know some parts or the important parts in the leaf, not in the leaf, in the flower. So if you look on here, as you know that for the flower, they have the pistil, they have the stamen, which is under the pistil, they have the stigma, the style, and the ovary. So all of these part in the pistil is really important for the uh, process of fertilization. Okay, and stamen, okay, they have the anther and they have the filament. So for this, where the male gametes will be dropped by, okay, so they have the petal also, they have the sepal, receptacle, and also the paddy cell. So I continue with seed development. So in my last slide, in first video, we are talking about the structure of the flower. Okay, where in the structure of the flower, we have the pistil, we have the stamen, and we have the petal. So if we talk about the reproductive structure, we need to focus on the pistil, where in the pistil we have the stigma, we have the style, and last we have the ovary. So the ovary is the most important part that you need to know if you want to talk or discuss about the reproductive system in plant. So in ovary, they may have or they may consist one or more ovules. So in this illustration is about one ovule. Okay, each of the ovule they have their own megaspore mother cell, where this megaspore mother cell okay, will be divide by mitosis to form four megaspore cells. So from four megaspore cell, okay, one okay, become the embryocyte when they do the meiosis process. Okay, another one it become a egg. So if you look on this ovule, you can see okay, one of them is labeled as egg cell. So one become egg cell. Okay, one become the embryocyte. So you have more two megaspore cell. So that two balanced megaspore cell so become the endosperm. So where endosperm in this ovule, okay, we call as a polar nuclei here at the center of the ovule. Okay, now we talk about the male reproductive structure. Just now we talk about the female reproductive structure. Okay, so for the male reproductive structure, we need to talk about the stamen. Okay, where in the stamen we have the anther and we have the filament. So in every anther, okay, they consist of microspore mother cell. For the female, we call as megaspore mother cell. However, for the male, we call as microspore mother cell, where similar things. Of course, this microspore mother cell will be divided okay, by meiosis and form the microspore. So this microspore, we call their general name as the pollen grain. Okay, where this pollen grain will be deposited onto the filament. Okay, and this pollen grain will be divided okay, mitotically to produce two cells, which is we call as tubule cell and generative cell. So I will explain to you later okay, in next slide regarding to this tube cell and generative cell. So in order to produce or to form the fertilization between the sperm cell and also the ovum, so this is the explanation. Okay, where they should be started with the male and it will be landing or it will be deposited okay, onto the female organ, okay, which is the story will be start with the pollen grain, which is microspore, okay, microspore cell, okay, are deposited onto the pistil. But the pistil we call as the stigma, so they will be deposited onto the stigma. Okay, so this we call as the process of pollination. Okay, then the pollen tube become to form the pollen tube. Because why? In the previous slide, I told you that the pollen grain will be divided. Okay, using the mitosis process, produce two cells, which is first is tube cell, second is the generative cell. So that tube cell will be formed the pollen tube. So when the pollen tube grow down okay, into the pistil, so it will be reached to the embryo site and also to the ovule. Okay, then what happened? The generative cell will be divided okay, to form two sperm. Okay, so these two sperm, okay, one will be fertilized the egg cell. Okay, so if you can see in the female structure, they have egg cell. So one sperm will be fertilized with that egg cell, and another one more sperm will be fertilized with the two polar nuclei, which is the endoderms. So this process, because they occur to fertilization, so we call as for the plant, they have the double fertilization. Alright, if you can't imagine what I told you just now regarding to the previous slide, so you can look on this illustration. Okay, where this one is regarding to the pistil, okay, where you can see here the pollen grain have been deposited onto the stigma, and the pollen grain will be divide, become the generative cell and also the tube cell. Okay, then first one, the the tube cell will be form the pollen tube, and when they produce a pollen tube, so this generative cell able to travel down and go to the ovule. So this generative cell will divide, become two sperm. Okay, so if you can see in the illustration, they produce two sperm, and these two sperm will be migrate down and penetrate at the opening in the ovule that we call as micropyle. Okay, so when they reach to the micropyle, so they able to do the fertilization, okay, double fertilization with who? With one sperm, okay, with one egg, and another one sperm will be fertilized with the polar nuclei or with the endosperm. So if you look on here, the statement last, okay, one of the sperm fertilized the egg to form the diploid zygote, okay, and another sperm fertilized two polar nuclei to form the triploid endosperm. So what function of the endosperm? Okay, to the embryo. Okay, in order they provide as a food source. Okay, in order the embryo able to grow and become the mature seed. Alright, so this one is still about the fertilization. So when the fertilization produce the zygote, so it will be resulting the highest number or increasing the cell division. So if increasing the cell division, so the number of cell also increase in the embryo and the endosperm. So this is the process where the first division of the zygote will be establish the polarity of the embryo, which is the upper part of the you can see the ovule will become the embryo, and the lower part will become the embryo sac. Alright, so this one is regarding to, in order we state that the seed have been starting the seed development is when the embryo, their shape is heart shape stage. So in this stage, they will be begin to appear their apical meristem. So here is when the embryonic develop. Okay, so here when the embryo when the, for example, fertilization have been produced and then produce the embryo, so the maternal plant will give the or will be allocate the nutrient, okay, to the embryo, okay, into the endosperm or cotyledon. So the endosperm and cotyledon, what you can say, they able to produce the nutrient or the resources that are necessary for the embryo to germinate. So they have two names of the seed here. Okay. If, for example, endosperm still remain, still have, okay, when the seed is matured, so that seed we call as endospermic. If, for example, at the seed at when for example when the seed matured, so the cotyledon enlarged and the size of the endosperm is reduced, so we call that seed as a non-endospermic seed. Okay, that is different between the endospermic with the non-endospermic seed. Okay, this is regarding to monocot seed. So the endosperm actually is surrounded by the aileron. So you need to not imagine, you need to always remember, okay, the endosperm always surrounded by the aileron. So this aileron is very important because they provide the embryo, okay, with the enzyme that able to break down the nutrient, and the embryo can get that nutrient. Okay, so because of that, in this segment they said that which provide the enzyme needed to mobilize the nutrient during the germination. So in this part we will explain to you more detail the process, okay, during next chapter. Okay, so for both monocot and dicot, okay, when they are become as an embryo, okay, and become matured, it will be as the dormant and desiccated stage, which is when they are matured. Okay, the seed always become dormant or desiccated. So when they need to, for example, germinate, they are waiting for the favorable condition. So if they get, I told you for in your first video, if they get enough oxygen, enough water, so they are able to germinate. And one more thing, okay, this seed, they surrounded by the hard seed coat. Okay, they have the coat, seed coat that we call as intergumen. Okay, intergumen actually is the seed coat that actually derived from the maternal tissue. Alright, so everything or every physiological process, they should be have the plant hormone involved. Okay, so same thing in the seed development. Okay, wherefore here they have four different plant hormones that play a big role in order to make the seed able to develop and able to germinate. So the name of the plant hormone is the cytokinin, auxin, gibberellin, and also the abscisic acid. So you need always to remember of these two graphs, okay, graph of the seed development and continue with the germination, because mostly in the test or in the final, they asking you about this statement. Okay, so for example, you look first on the seed development. Okay, you can see that the CK is the cytokinin. Okay, the concentration is increased, so means the higher is during the early, okay, the early stage of the seed development, right? And then after that, okay, you can see GA means gibberellin, and IAA is means the auxin. So you can see that at the mid, okay, at the middle part of the seed development, so these both plant hormone increase and the cytokinin concentration is decreased. So this means where the cell have the enlargement process, which is they have a lot of the or increase the number of the cell division in the embryo. Okay, and late, okay, at the seed development, you can see they have the ABA. So ABA is the abscisic acid. So abscisic acid concentration peak, okay, when the seed matured, and this contribute to the dormancy and desiccation of the embryo. And you can see that before the seed able to germinate, they may enter the quiescence stage, which is this stage we call as resting stage for the seed. Okay, as I told you just now, for the quiescence, they are the resting condition. Okay, when the matured seed they impose to the dehydration, means that seed really have a very low concentration of water. So in order the plant able to breaking the quiescence and able to germinate the seed, they should be influenced by these three factors, which is they need to have adequate water, so means they need to have enough water to rehydrate the seed. Okay, they need to have enough oxygen for the respiration of the plant. Okay, and they need to have the optimum temperature, which is in range 25 to 45 Celsius. So they may germinate the matured seed after that. Alright, so the last one is regarding to the difference between the quiescence, okay, and the dormancy. Okay, because if you look on the graph, okay, we call as quiescence. So actually, for the quiescence and uh, dormancy, they have a similar meaning, which is this means they are the resting condition for the matured seed. However, the condition is different. Okay, which is for the squamous, you can see they are the state of the suspended growth of the embryo, okay, or a resting condition. And in order we able to break the squamous stage, we just only expose that matured seed to the favorable condition. Means we just put water, have enough oxygen and optimum temperature so they able to germinate. However, it's different to the dormancy. They are similar condition, means they are the resting condition. However, in order that matured seed able to germinate, they require a special event, or they need to trigger by a special treatment, okay, which is the treatment with a fire or clarification or with cold treatment. So with these all types of the treatment or special event, we able to uh, break the dormancy of the seed and it able to germinate. So until all these events occur, so the seed is prevented from the germinating. That's all, student. Thank you.
Lately, I've been thinking, I want you to be happier. I want you to be happier. When the morning comes and we see what we've become, in the cold light of day, we're a flame in the wind, not the fire that we begun. Every argument, every word we can't take back. Cause with all that has happened, I think that we both know the way that the story ends. Then only for a minute I want to change my mind, cause this just don't feel right to me. I wanna raise your spirits, I want to see you smile, but no, that means I'll have to leave. No, that means I'll have to leave. Lately, I've been thinking, I want you to be happier. I want you to be happier. When the evening falls And I'm left there with my thoughts and the image of you being with someone else. Well, it's eating me up inside. But we ran our course, we pretended we're okay. Now if we jump together, at least we can swim far away from the wreck we made. Then only for a minute I want to change my mind, 'cause this just don't feel right to me. I wanna raise your spirits, I want to see you smile, but no, that means I'll have to leave. No, that means I'll have to leave. Lately, I've been thinking, I want you to be happier, I want you to be happier. So I'll go, I will go. So I'll go, I will go. Lately, I've been thinking, I want you to be happier, I want you to be happier. Even though I might not like this, I think that you'll be happier, I want you to be happier. Then only for a minute I want to change my mind, 'cause this just don't feel right to me. I want